last year was a big year, Sam, and I just wondered if you would take a minute to provide investors on the call, what should we take away right now regarding CRISPR? What should investors be keenly aware of?
Yeah, thank you, Tony, for having us at this conference. It's always great to see your notes. You've been following us for a long time, and during that time, pleased to tell you that, as you know, that we've made a lot of progress. We've grown from a very small company to being the premier gene editing company today with over 400 employees. And more importantly, last year was a landmark year for us. On the heels of Dr. Charpentier and Dr. Doudna winning the Nobel Prize, we showed data for our sickle cell and beta thalassemia programs where 10 of 10 patients were effectively cured after treatment with CTX001. And the side effects were all similar to what you see with busulfan autologous transplants. So I think that was a major data update. I think we also announced data for our CTX110 program in immuno-oncology, where we showed that a CAR T made from a healthy donor source can actually kill the cancers and provide a very high response rate in DLBCL, which is one of the worst forms of non-Hodgkin's lymphoma. And while there's more data to come, I think we're very encouraged by what we saw in that first data release for CTX110. 2021 promises to be an even more important inflection point for us. We have several different data catalysts. We have additional data from our CTX001 program, and hopefully we hope to get regulatory clarity as well in terms of what's required for filing, so we can bring that therapy to market. For all our CAR Ts, all 3 CAR Ts, we have data coming up this year that would show how these CAR Ts are doing, the respective indications they're being developed in, and also, and we're going to talk about it, but we're going to bring our first regenerative medicine program to the clinic, or file for an IND or CTA by the end of this year. So quite a bit happening for us from a data catalyst standpoint, but overall, I think we're very pleased with the ability to have a very talented group of people at CRISPR doing all this work, a market cap that provides us a very strong balance sheet position, and ultimately a set of portfolio choices which allow us to scale this company to a $20-25 billion company in the near term, should we get positive data.
Yes, Sam, thank you for that. And just from a historical perspective, I remember many years ago when CRISPR was actually formed, and some of the early thoughts around the clinical programs were simply in beta thalassemia and sickle cell disease. Those are clear, and you highlighted them. And then the move over to allogeneic CAR Ts, I think, was sort of underrepresented by the investment community, and you clearly proved that you can solve that problem. I wanted to focus on something that I think respectfully hits the shortest straw, and the reason for that is I often believe that if you're successful at hiring really good people, all of a sudden a group of people, or maybe one, they're clearly in tune with the literature, they're in tune with the progress at the organization, but they all of a sudden think about, well, hey, I wonder if we could address applications in this. What would we need to do in order to make that happen? And clearly one of those programs you mentioned in regen medicine, but in type 1 diabetes. So if I think about your relationship with viacyte back in, and I think this was back in 2017, Sam, there's been a good bit of progress. It's taken some time, but a good bit of progress that's been made. And I don't think that the street's really paying a lot of attention to your regen med program. And I'm being respectful. We don't have any clinical data, so we don't know. But there's some really interesting biology that's gone into why it might be successful. And I just wondered if you would touch on this program, and in particular, because it's potentially in type 1 patients, and you're moving forward with the trial, I assume many of those patients are going to already be on insulin. So touch on what does that mean if you're really trying to solve their insulin problem, and yet they may be on insulin when they enter the study.
Yeah, thank you. You are right to draw parallels to our pipeline in 2017, 2018 timeframe, Tony, when all the focus was on hemoglobinopathies, and we got very little credit for allogeneic CAR Ts, which today have equal value in our enterprise value, if not more, relative to hemoglobinopathies. And I recall the conference you did actually in New York at that time, which highlighted the power of allogeneic therapies and the possibilities. And I think people have now recognized the potential of allogeneic therapies. I think we're in a similar position with regen med. What I'll point out is, the first question is why regen med for CRISPR? And what you know is in the last 20 years, there's been tremendous progress in directing the fate of cells. You can start with a stem cell and direct its fate to any cell type that we want or dream of. And whether it's liver cells, brain cells, heart cells, because ultimately we all as humans came from that one single cell. And in 2012, there was a Nobel Prize for Dr. Yamanaka around the discovery and development of directing the fate of cells from induced pluripotent stem cells to other cell types, but more importantly, the ability to create induced pluripotent stem cells. And this type of approach works. There have been a number of clinical trials where people have taken hair cells or skin cells from patients, reprogrammed them to corneas or retinas, and use them in patients surgically, and it works to regenerate any organ of interest. But it's very difficult to do on a per patient basis. Right? You're doing it, it's going to cost you $1 million, $2 million, but it's going to take you 6 months to do for each patient, which is not really commercially viable. That's where CRISPR comes in. So if you now then say, if you can use CRISPR, the technology, to edit these cells to make them stealth, you can go with the master cell bank and fulfill the needs of every patient. It's going to, in 10 years, we're no longer going to have this wait for kidney transplants or liver transplants. I think you're going to have artificially produced organs just to transplant people and overcome the failed kidneys or failed livers using regen med in a way where there's no rejection by the patient's body. So that's one force, which is we wanted to play in regen med given that 1 + 1 = 3, which is regen med plus CRISPR can provide tremendous value, and there's synergy there. The second thing is, if you look at type 1 diabetes and diabetes in general, it's a tremendously high unmet need. You've seen all the news around insulin and what the impact of that to the system as well. So there's unmet need not just from a individual patient standpoint, from a system standpoint. And what's interesting is about 20 years ago, around the dot-com boom era, there was this thing known as Edmondson protocol that was discovered, which was that you can take cadaveric islet cells that are the cells of the pancreas and just simply inject patients together with and immunosuppress the patients, and those cells can function as your pancreas and produce insulin in response to glucose. And all those patients, almost 500 patients, who were sort of transplanted or injected with these cells, cadaveric cells, and they all did pretty well, even though they were they had severe disease, severe type 1 diabetes. And when we say did well, they didn't have any glucose excursions, either hypoglycemia or hyperglycemia, and their disease was controlled. Now, it's very hard to find fresh cadaveric cells. It's in short supply. And this notion then led us to say, if you can regenerate cadaveric islet cells, not from a cadaver, but from a pluripotent stem cell or embryonic stem cell, then all of a sudden you have all the supply you need to inject patients with these cells. And if you can use CRISPR to make them stealth, then you have a solution that can apply not just for type 1 diabetes, but also for type 2 diabetes. And maybe one day the whole insulin market disappears if this works. And so it's a very powerful idea. And we were fortunate to find a partner like viacyte, which is based in San Diego in the La Jolla area. And they've been around for almost 20 years and developed the protocol around taking stem cells and differentiating them into islet cells, or at least precursors of islet cells. And these precursors can infect cells. And if you take these precursors of islet cells, put them into patients, and they've done that. They've actually dosed 20 plus patients with these cells. They do differentiate fully into islet cells in the body of the patient and produce insulin in response to glucose. So it's a very powerful idea with clinical validation from the Edmondson protocol. And we're very excited to embark in the clinic with this program either later this year or early next year based on when exactly we file the IND and CTA.
Yeah. So, but Sam, I think one of the points to highlight also is the fact that you're doing this in a non-immunosuppressed patient, right? I mean, this is kind of one of the beauties is that somebody doesn't have to walk around taking immunosuppressant drugs for the rest of their life because their T cells are going to attack the tissue that you, in theory, implanted into them. So I think it's pretty unique to think that not only could you solve the problem, but you remove some of the, let's say, hazard around future immunosuppression.
And that's why CRISPR is so powerful in this concept, which is the need for immunosuppressives in these patients is that if you have foreign cells injected into patients in the Edmondson protocol, they will get rejected by the host immune system, primarily by T cells. And if you immunosuppress these patients, then they don't get rejected and they can do their work. Now, immunosuppression is a hazard, especially as we live in the times of the COVID pandemic. Immunosuppressing patients for a long time exposes them to all sorts of infectious diseases and could lead to several comorbidities and oftentimes death. So if you can eliminate the need for immunosuppression, and the way we do that is by making edits on these cells that prevent rejection by the host immune system. At least a couple of the edits we've talked about in conferences. One is a beta-2M edit, which we also have in our immuno-oncology programs, where we knock out the beta-2M to essentially eliminate the MHC class 1 complex on these cells, which prevents the T cells, the host T cells, from recognizing and rejecting these cells in the body. So that's something we've studied extensively, and we believe has value here in the regen med context. The other edit we've talked about is a PD-L1 knock-in. We've all seen the power of the PD-1 drugs like Keytruda, and I think knocking in PD-L1 also prevents this T cell attack. In addition to that, we have 3 more edits that we make to these cells to improve cell fitness, improve their proliferation, and ultimately increase their ability to be stealth in the face of the host immune system.
And so these cells get implanted in a I'm going to call it a sac, but there's a membrane around them. It's the core membrane. I just wondered if you would speak about that for a moment, because my guess is if you just put the cells, if you planted the cells into the pancreas, they probably don't stay. They do need to get electrolytes and other types of and have some blood supply. What does the core membrane do to them?
Yeah, I think that the notion of putting them into a sac or membrane or device, whatever we call it, is several folds. One is you're keeping the cells all in one location, one device, where if you need to retrieve the cells, you need to retrieve them. You just take the device out and put a new device in, right? Let's say you end up with these cells that have a 5-year life. Every 5 years, you can just take this old device out and put a new device in to serve as artificial pancreas. The second thing is, I think these cells do work together if they're all in one, in one cluster. And what we want to do with these devices is to make sure that they have enough nutrients and are able to survive and proliferate in the devices. And the core membrane comes in because not only does it have pores which allow vascularization of the device to provide nutrients to the cell, they also prevent foreign body reaction or foreign body response. And if you put them in other devices, you get these giant cells that envelop the devices and just cover them up. It's our body's way of rejecting the device, if you will. And you've seen that with other types of implants before. But these core membranes that are originally based off of PTFE and are relatively the mix of hydrophobic and hydrophilic that makes it very attractive, which is what you see in ski jackets. I think on one hand they can absorb all the sweat and allow aeration. On the other hand, you easily tap the snow off your jackets, right? I think it's a special material. And I think we use something like that here to prevent that foreign body reaction on these devices and ensure vascularization. I think it may be that the device in itself can actually give significant stealth capabilities to these cells and reduce the need for immunosuppression. But I think that combined with the editing is a very powerful combination where you've localized the cells, you've created the retrievability of the cells, and you've enhanced their survival and proliferation without causing a foreign body reaction, and ultimately enhanced the stealth nature of the cells as well.
Thank you for that. I appreciate the lesson and the work. I want to come back to hemoglobinopathies full circle, Sam, because you made a statement earlier, and I've heard you say before, as it relates to what is the FDA going to make you do in this particular area. So I want to spend a minute on commercial preparedness, if you will. I don't know how much that phrase takes up room in your brain, but it's not a trivial discussion. I suspect it might take up a lot of time because you always want to know when do we exert capital to this program or that program so that we get ready if in fact we are to move forward into the commercial market. So could you spend a few minutes there?
Yeah, I think I'll start off by saying that we're very pleased to have Philippe Drouet join us as Chief Commercial Officer. He joined us last month, and this is an important signal in terms of the phase of growth of our company. And the commercialization capabilities we want to build are obviously for CTX001, it's in the U.S., because we're leading commercialization in the U.S. for that program. But for all the immuno-oncology programs, it's global. I think we want to ultimately be fully integrated and have those capabilities, whether it's in the U.S. or in European markets, and then we'll evaluate Asia or the rest of the world as we develop the programs further. So commercialization and the commercial infrastructure build is actually very important for the company. And there are a few thoughts here that I'll talk about. One is, it's really important while you build your commercial infrastructure that you continue to maintain the culture of the company and maintain that sustainable innovation engine that got you here in the first place. This has been a pitfall for many companies that they get so engrossed in commercialization that they forget about value creation from research and development. That's been the raison d'etre for all companies like us. The second notion is getting a launch done successfully early on is very important. We've all seen companies go through this value depth. You have tremendous promise and excitement before approval, and oftentimes you've seen stocks trade down upon approval as small companies invariably struggle a little bit with some of the early launches, which ultimately catch it, in a good place. But this notion of starting strong with the launch early is very important, especially in hemoglobinopathies where it's competitive. And I think we're doing everything we can to be in that place so that we get, we're prepared for whatever the regulatory filing timeline is, and ready to go from a commercial infrastructure standpoint. But that extends not just to, from a typical commercialization standpoint, but also manufacturing, supply chain, and sort of the patient experience that's necessary to successfully sell a drug like CTX001. So
does that imply that we'll hear more announcements, let's say, on the manufacturing front, or will you make more statements onto your ability, or at least your belief that you've got sufficient capacity?
Yeah, no, I think it's kind of linear in terms of how we're increasing capacity. There's not, it's not one or two moves that we're making that all of a sudden